Assalamu alaikum warahmatullah. In this video we are going to keep talking about the reaction of cellular respiration. So as you know right now, cellular respiration has three main phases. The first phase is glycolysis reactions, and second we have the Krebs cycle, and the last phase, which is the oxidative phosphorylation or the electron transport chain. In this video especially, we are going to be talking about the oxidative phosphorylations. Well, before getting into it, let's summarize the previous reaction by counting the number of the energetic molecule we got from the previous reactions. And let's start with the output of glycolysis reaction. So if you do remember, in glycolysis we only produced 2 ATP molecule and 2 NADH+. For the second phase, we have actually two steps: the preparation step, then the Krebs cycle reaction. So for the preparation step, we produced 1 NADH+ for every single pyruvic acid. So since glycolysis we've produced 2 pyruvic acid, we will have the production and the preparation step 2 NADH+. For the Krebs cycle, we've produced 3 NADH+, 1 FADH2 and 1 ATP for every pyruvic acid. So we actually have to multiply all this by 2. So in the Krebs cycle we produce 6 NADH+, 2 FADH2 and 2 ATP. So if we count all the ATP molecule produced in all these reactions, we will find that we've produced only 2 ATP in glycolysis and 2 ATP in the Krebs cycle. So only 4 ATP. But actually we've produced so many electron transport molecules such as NADH+ and FADH2. So in general, we've produced 10 NADH+, 2 FADH2 and 4 ATP. So you can see over here the huge differentiation between the number of ATP molecule and the number of the electron transport molecule. The problem is our cells are going to be using only those ATP. They do not use FADH2 and NADH+. So what our cells are doing, they are going to take these electron transport molecule and convert them into ATP so that our cells can use them. Now the question is how our cells are going to convert NADH+ and FADH2 into ATP, and where that is going to happen. And at this point the oxidative phosphorylation will get involved, because in this phase we will see the destiny of all these electron transport molecule and how they will be converted into ATP molecule. Now let's start with the question where that is going to happen. And so as you can see over here, this is a section of the mitochondria. This structure over here is the outer membrane of the mitochondria. This space is going to be the intermembrane space, and this structure is going to be the inner membrane, and the space inside of the mitochondria is going to be the matrix. So actually the electron transport chain happens along the inner membrane of the mitochondria. So if we take a look into this inner membrane of the mitochondria, we can see its chemical composition. So over here we have the phospholipid bilayer, then we have this structure over here that we are going to call proteins, and especially transmembrane protein. and we will call each one of them a complex. So this is the complex number 1, complex number 2, number 3, 4, 5, and 6. So T over here refers to the proteins that we can find in the inner membrane of the mitochondria, and that they form what we call the respiratory chain. And over here at the end, we can find the famous molecule of producing ATP, which is the enzyme ATP synthase. So now the question is how this electron transport chain actually works. Initially, in the matrix, we saw that we have the Krebs cycle reaction. So we will have the production of NADHH plus and FADH2. Let's start with NADHH plus. We said that this molecule is going to be converted into ATP, and to make that happen, NADHH plus is going to be reoxidized into this inner membrane of the mitochondria, and especially in the complex number 1. So NADHH plus is going to be oxidized into NAD plus 2 electrons and 2 protons. For FADH2, it's going to be reoxidized also, but not in complex number 1. It's actually going to be reoxidized in complex number 2. So FADH2 is going to be converted into FAD plus 2 electrons and 2 protons. So as you can realize from both reactions, we will have the releasing of protons and electrons. For protons, we really don't have a problem with them because they can be free in the matrix. But the problem is for the electron. The electron, they are never going to be free. So how can we solve this problem? Let's start initially with NADHH plus. So in the oxidative reaction, we will have the releasing of the electron and protons. For the electron, they are going to be flowing from complex number 1 into complex number 3, then complex number 4, then complex number 5, until the complex number 6. So this complex number 6 is going to give these electrons into oxygen. Oxygen that we are going to find into the matrix of the mitochondria. So what actually is going to happen is oxygen will receive the electron and try to get some protons from the matrix, and it's going to be reduced into water. So if you do remember in the previous video, we said that cellular respiration is aerobic pathway, so it requires oxygen that is going to be involved into the electron transport chain as the final electron acceptor. Now this electron flow through these proteins is going to release energy, and the energy released is going to be used to pump the protons from the matrix into the intermembrane space. How? This is what we call active transport. So we need energy and we need a pump. So for NADHH plus, these protons released are going to be pumped by the complex number 1 and complex number 4 and the complex number 6. The pumping of protons will create an electrochemical gradient, which means we will have proton accumulation into the intermembrane space of the mitochondria, which is going to make the concentration of protons over here increase. And the other side, the protons concentration is going to decrease into the matrix First, because we have the pumping of protons using the energy coming from the flow of electron, and second, because we have here the reduction of oxygen into water, and this reaction requires protons. So this is what we are calling the electrochemical gradient. It's only the differentiation of concentration between the intermembrane space and the matrix. Now, for these protons that are in the intermembrane space, they are going to search to back in the matrix, and the only way they can back in is through this molecule over here, which is the ATP synthase. So what happens is this ATP synthase will allow the flow of protons through this channel back to the matrix, and when that happens, ATP synthase is active and powers the production of ATP. So over here, let's talk about this proton. It's going to flow to the matrix, and this flow is going to release the energy. This energy will be used to make a chemical bond between ADP molecule and this phosphate group to make ATP. And this reaction over here is what we've called phosphorylation. So we started from the oxidation of NADHH plus, and we end up by the phosphorylation reaction. So this is why we call all this phase oxidative phosphorylation. Oxidative because we are going to start by the oxidation of NADHH plus and FADH2, and phosphorylation because we will end up by this reaction of making ATP. And if you realize another thing, through these reaction we've converted NADHH plus molecule into ATP. So if you do remember, we said that protons are going to be pumped by complex number 1, number 4, and number 6. So we will consider that the reduction of NADHH plus is going to pump 3 protons. And for every protons coming back into the matrix through the ATP synthase, we will have the production of 1 ATP molecule. So for these 3 protons, we will actually have the production of 3 ATP molecule. In other words, we will take every 1 NADHH molecule as the equivalent of 3 molecule of ATP. Now back to FADH2. So actually we will have the same steps, but only with few differences. So for the differences, the first one we said is the oxidation of FADH2 is going to happen in the complex number 2. Then we will have the electron flowing through the respiratory chain, and that they will end up accepted by oxygen that is going to be reduced into water, and the flow of these electrons is going to release energy. This energy is going to be used to pump the protons. Now the protons coming from the oxidation of FADH2 are going to be pumped through complex number 4 and complex number 6. So we will have only 2 protons in the intermembrane space. Actually we have a lot, but we will just keep in mind only 2. And this is going to produce the electrochemical gradients. So these protons, they are going to back into the matrix through the ATP synthase, and from every proton coming back into the matrix, we will have the releasing of energy that is going to be used to produce ATP. So we have here 2 protons. When they will back in the matrix, they will produce the 2 ATP molecule. So what we are going to do is we will consider every FADH2 molecule as the equivalent of 2 ATP. So that's it. This is how we convert the electron transport molecule into ATP, so that our cells are going to use them. Now let's try to count how many ATP molecule we will get from this metabolic pathway. Summary of cellular respiration. And the first phase we said it's glycolysis. So in glycolysis we've produced 2 ATP and 2 NADHH plus molecule. So every 1 NADHH plus molecule in the last phase is going to be the equivalent of 3 ATP. So actually we will have in glycolysis the production of 6 ATP in addition to these two. So in glycolysis we will have the production of 8 ATP molecule. For the next phase, we have the preparation step, which is from the pyruvate molecule into the acetyl coenzyme A. We will have the production of 2 NADHH plus molecule, and each NADHH plus molecule is the equivalent of 3 ATP. So it's just like if we have a 6 ATP after the oxidative phosphorylation. And for the Krebs cycle, we've said that we will have the production of 6 NADHH plus, so we will multiply 6 by 3 ATP, so we will have the production of 18 ATP. FADH2 we have 2, that when they will be reoxidized into the electron transport chain, they will give 2 ATP, so we will end up by 4 ATP. And the last then we have the 2 ATP that are not going to be converted. Now, in total of all these reactions of cellular respiration, we will end up by producing 2 plus 6 ATP plus another 6 ATP plus 18 ATP plus 4 ATP plus 2 ATP. So we will have in total 38 ATP, and this is the output of cellular respiration. Before ending this video, the last thing I want to mention is the global reaction of cellular respiration that you should put in mind, and this is the global reaction. So over here we have C6H12O6, which is the chemical formula of glucose molecule, plus oxygen, that they are going to be converted into carbon dioxide and water and 38 ATP. And in other words, we said that cellular respiration is the total breaking down of glucose in the presence of oxygen. So in the end products, we really don't have any more molecule that they still contains energy. We totally broke down the glucose molecule. Now, all these reactions happen for the reason is to get energy, and we will have 38 ATP from every glucose molecule that enters our cells. Now we have here the production of water. This water is coming from the oxidative phosphorylation reactions, especially from the reduction of oxygen into water. For the carbon dioxide over here, we will have it from the Krebs cycle. So this is the carbon dioxide that we release when we do respiration. So hope this video was helpful, and see you in the next one.